questions based on the major functions of B cells, including humoral immunity, antigen recognition and presentation, somatic hypermutation, antibody production via plasma cells, immunologic memory, memory B cells. Each question includes 5 answer choices with full explanations. Question 1. Core function. Which of the following best describes the primary role of B cells in the immune system? A. Phagocytosis of pathogens and debris. B. Direct killing of virus-infected cells via perforin. Secretion of cytokines to activate macrophages. D. Production of antibodies as part of humoral immunity. E. Antigen presentation via MHC class I. D. Production of antibodies as part of humoral immunity. Explanation. B cells are responsible for humoral immunity, mainly through antibody production. Upon activation, they differentiate into plasma cells, which secrete specific immunoglobulins. Why others are wrong? A. Done by macrophages and neutrophils. B. CD8 plus T cells and NK cells perform direct cytotoxicity. C. T helper cells, CD4 plus, primarily release cytokines. E. B cells present antigen via MHC2, not I. Question 2. Somatic hypermutation. The process by which B cells improve their antibody affinity after activation is best described as A. Class switching. B. Somatic hypermutation. C. Antigen drift. D. Central tolerance. E. Complement fixation. B. Somatic hypermutation. Explanation. Somatic hypermutation introduces point mutations in the variable region of the B cell receptor to increase affinity for the antigen. Why others are wrong? A. Class switching changes isotype, e.g. IgM-IgG, not affinity. C. Antigenic drift refers to viral mutations, not B cells. D. Central tolerance deletes self-reactive lymphocytes. E. Complement fixation is a function of antibodies, not B cell maturation. Question 3. Plasma cell differentiation. After antigen stimulation, a B cell differentiates into a plasma cell. Which of the following best describes the function of plasma cells? A. Engulf and digest pathogens. B. Present antigen to naive T cells. C. Produce and secrete large amounts of antibody. Kill infected cells using perforin and granzyme. E. Inhibit autoimmune responses via IL-10. Answer. C. Produce and secrete large amounts of antibody. Explanation. Plasma cells are terminally differentiated B cells that act as antibody factories. They secrete large amounts of immunoglobulin to neutralize pathogens. Why others are wrong? A. Phagocytes do that. B. APCs like dendritic cells activate naive T cells. D. CD8 plus T cells and NK cells. E. Regulatory T cells inhibit autoimmunity via IL-10. Question 4 memory. Which of the following features of B cells explains long-lasting immunity after vaccination? A. Polyclonal antibody response. B. Central tolerance. C. Memory B cell formation. D. Isotype switching. E. Passive immunity. C. Memory B cell formation. Explanation. Memory B cells persist after the initial immune response and respond rapidly to subsequent exposures, accelerated and more effective response. Why others are wrong? A. Polyclonal response diversity, not persistence. B. Central tolerance eliminates self-reactive clones, not memory. Isotype switching changes antibody class, not memory. E. Passive immunity, externally acquired antibodies, e.g. IgG from mother. Question 5. Antigen presentation. Although primarily antibody-producing cells, Bs can also present antigen. What type of MHC molecule do B cells use for this function? A. MHC class I only. B. MHC class II only. C. CD1 molecule. D. MHC class I and CD8. E. beta-2 microglobulin only. B. MHC class II only. Explanation. B cells act as antigen-presenting cells by displaying processed antigens on MHC class II to CD4 plus helper T cells. Why others are wrong? A. MHC1 is on all nucleated cells, but antigen presentation to CD4 plus requires MHC2. CD1 presents lipids, not peptides. D. CD8 interacts with MHC1. B cells present to CD4 plus via MHC2. E. beta-2 microglobulin is part of MHC1, not 2. Let me know if